In this video, we're going to run through one of the required practicals where we investigate the effect of light intensity on the time taken for chloroplasts, or a suspension of chloroplasts, to decolorize a solution known as DCPIP. So what we're going to do here is we're going to run through the background science behind this experiment and then hopefully watch together a demonstration of the investigation being carried out. So things to bear in mind then. In photosynthesis, we know that we have to take hydrogen atoms in order to reduce glycerate 3-phosphate, or G3P, to triose phosphate. And this is done in a light-independent reaction. But these hydrogen atoms have come from the light-dependent reaction, so the series of reactions which require light energy. So this happens when we have photolysis of water, or the splitting of water in the light-dependent reaction. We have an electron released during photolysis of water, which is used to replace an electron that has already been lost by chlorophyll. This happens because the light energy is able to excite an electron. So in the chlorophyll, we have excitation of electrons using light energy, the release of these electrons, and then we are able to have a coenzyme called NADP, different to NAD in respiration, that is able to then become reduced because it accepts these electrons, and it also at the same time accepts a proton released by the water splitting. We know that then the reduced NADP, or NADPH, is able to move through or diffuse to the stroma, and there it's able to donate its hydrogen, so it's able to become oxidized in order to reduce glycerate 3-phosphate to triose phosphate. Okay, so what we're going to do is we're going to take advantage of the light-dependent stage. So instead of allowing the chloroplasts in our suspension, instead of allowing them to reduce NADP in the light-dependent stage, we're going to provide them with this solution called DCPIP. Now, DCPIP is a very rich blue colour. Now, when that is mixed in with the chloroplasts, what we will have happen instead is that a lot of the electrons will be transferred or accepted by the DCPIP instead of the NADP. So our DCPIP should become reduced when the chloroplasts are carrying out the light-dependent stage of photosynthesis. Why is this important? Well, DCPIP, when it becomes reduced, turns from a blue colour, so when it's oxidized it's blue, to colourless. So we are going to have a visible change in colour. So as you can see from this equation below, oxidized DCPIP is blue. When it becomes reduced, it becomes colourless. And it becoming reduced should be due to the presence of the chlorophyll, which is in the chloroplasts, and the progression of the light-dependent stage. Okay, so we can roughly estimate the rate of photosynthesis by how quickly the DCPIP loses its blue colour. Faster photosynthesis, or faster light-dependent stage, we should have more photolysis of water, or faster photolysis, a faster rate of electrons being emitted, and so the DCPIP should become reduced more quickly, and we should have the colour change more quickly. Hopefully, if we have a lower intensity of light, which is what we're going to investigate the intensity, we should have the converse of that. So less light energy available, less photolysis, less emission of electrons, or a slower rate of them, and a slower rate of change, so a slower colour change. So what we're going to do is we're going to hopefully prove that it's the DCPIP that is reduced by the chloroplasts themselves, and also that it is the light that is causing this reaction. And we're also going to be investigating the effect of the light intensity on the time taken for the DCPIP. So how does light intensity essentially affect the rate of photosynthesis? So we're going to have a solution of DCPIP, we're going to have a suspension of chloroplasts, we're going to have a lamp, we're going to have many test tubes, and a stopwatch or a timer. Now in this, interestingly, although we are judging this reaction by a visible change in colour, we're not going to use a colorimeter. we are going to use our own judgment. So there is a lot of room for human error in this. Things that we are going to attempt to control in this investigation, so factors that could affect the results that we are going to try and minimise the effect of. We are going to try and minimise the effect of temperature. We know that photosynthesis involves a lot of enzyme catalysed reactions, and we know that therefore temperature can have a really significant effect on it. So we are keeping all our key solutions chilled. The chloroplasts are being kept in a cold isotonic buffer. The DCPIP is chilled, and the water that we're going to use is also chilled. We're going to make sure that we use the same volume of chloroplast extract, and we're going to select appropriate equipment for measuring that volume. So you will see in the video we have different choices of syringe sizes. We need to consider things like tolerance and percentage error in order to select the most appropriate piece of equipment. Same thing for the volume of the DCPIP solution, which is the most appropriate syringe size. Wavelength of light. We know that chlorophyll absorbs different wavelengths in different amounts, so we're going to use the same source of light, so the same lamp. And then also we are going to ensure that our chloroplasts, which are suspended in solution, that solution is a cold isotonic buffer, and why that is important. So what will now follow are a few video clips of the practical being carried out. So things to bear in mind: watch carefully, consider the methodology, the reasoning behind all the steps, why we have the controls that we are using. So why do we use tube A and tube B? What is the purpose of tube C? Why is everything chilled, as we've already talked about previously? Are there any safety aspects we need to consider? And also spot the deliberate mistake. So within the first few seconds, I definitely do not refer to photosynthesis as photosynthesis. So you can pat yourself on the back when you hear me say the complete wrong term for that. So sit back and enjoy. In this video, we're going to run through one of the required practicals, which looks into the effect of light intensity and changing it on the time taken for chloroplasts to decolorize a solution called DCPIP. So for this investigation, let's just run through what apparatus we have. So we have got an extract of chloroplasts in here. Now this is being kept on ice, so it's being kept cold. Consider why it would be of benefit to keep these chloroplasts cold. Okay, we have also got DCPIP solution. Note the colour of it, it's quite an intense blue. It is also, not that you can tell, but it is also cold. We've also got some distilled water. This is also cold. Everything's cold. There's a bit of a theme here. Okay, we've got a selection of test tubes that we're going to use for our different runs of the experiment, and obviously we've got them in a rack. We've also got a selection of different syringes. Now consider as well, when we're going through this, why we have got these different sizes. So for example, this syringe goes up to 1 centimetre cubed only. That's the maximum volume that it can measure out, whereas these larger ones are able to go up to 5 centimetres cubed. Okay, so we have a selection of those there. We've also got a piece of aluminium foil. We've got a lamp, a very rudimentary lamp. We've got a marker pen, and we've got a timer. We've also got a ruler. So we're going to hopefully piece these all together and run through and see how to carry out this investigation. So hopefully from previously in the video you understand what it is that we are doing or why we are using DCPIP for this. So DCPIP, quite similar to methylene blue, is a solution that is able to change colour. So we get a visible change with this investigation. So what we are hoping is that the chloroplasts, we enable them to undergo the light-dependent stage of respiration, so with light, and hopefully this will result in electrons being lost from the chlorophyll, the photolysis of water, and what would normally happen is that NADP would be reduced. Now, by providing the chloroplasts with DCPIP, we are hoping that instead it's the DCPIP that becomes reduced. Now, when DCPIP becomes reduced, it becomes colourless. So we are looking for a change from this blue to colourless. Now, obviously, we are going to be mixing this with chloroplasts and therefore chlorophyll So these are going to be quite green. So we're not looking for something to go blue to colourless. So this is why we're going to have a comparative tube. But instead we're going to be looking for a bluish green, okay, changing to more of a pure green. Okay, so we'll run through the steps involved. What we're going to do first of all is we're going to set up a couple of controls. So we're going to take these test tubes and we are going to label one of them as A, okay, and the other as B. Okay. Now what we're going to do is in tube A, we are going to add 5 centimetres cubed of the DCPIP solution, 1 centimetre cubed of water, and 1 centimetre cubed of chloroplasts. But the important bit here is after this, we are going to wrap that tube with this aluminium foil. The idea for this is that we are going to exclude all light from getting to the chloroplast. So we are hopefully going to stop the chloroplasts from carrying out the light dependent reaction. The logic being that without the light we should not get any of the reduction happening of the DCPIP, so we shouldn't really get a result. And this is going to prove to us that it is the light that is causing the reduction of the DCPIP, or the light dependent reaction, and nothing else. So that's going to be our first one. In the second tube, tube B, we're going to place 5 centimetres cubed of the DCPIP. We're going to have 2 centimetres cubed of water and no chloroplasts. This is going to prove to us that it is the chloroplasts that are causing the reduction of the DCPIP, so causing the colour change. That it's not just the DCPIP spontaneously changing colour, or it's not the effect of the water. Okay, so again, we've got these two controls. So I'll set those up now. So we take our DCPIP. At the moment, we need to decant that into a smaller beaker so that it is easier for us to use. Okay, and then it's 5 centimetres cubed. So as you said, this one only goes up to 1. We'd have to use that 5 times. I mean, we could do, but not brilliant. Whereas this one goes up to 5, so it's more appropriate. So we'll take our 5. Okay. We're adding that to tube A. And then we need 1 centimetre cubed of the water. So again, we could use this larger syringe that goes all the way up to 5, but it would be far better for us to use the 1 centimetre cubed. Okay, add that in. Remember to keep your syringes with one another so we don't get any cross-contamination. And then finally we're going to add 1 centimetre cubed of the chloroplast solution. So you can see it is a wonderful green colour. Okay, and it's being kept on ice. And then we need to quickly wrap this up in tin foil. So we are hoping to get absolutely no gaps, really good seal on it, so that we don't have any of the light dependent stage happening. So that is our first control, and we're hoping that when we come back to that we won't see any colour change. Second one, remember, we are keeping the volume the same, so we had 5 of the DCPIP, we had 1 of the water and 1 of the chloroplast solution in here, so a total of 7 centimetres cubed. We don't want the chloroplasts this time, so instead we're going to double up on the water. So another 5. And this time, rather than using 2 rounds of the smaller syringe, this one only goes up to 1 centimetre cubed, we can use a slightly larger one, a fresh one, that goes up to 5, because then we can measure to 2 quite easily. Okay, and we are not putting any chloroplasts in there because this is going to hopefully show us that it is the chloroplasts that are having effect. So those are our controls now set up. What we will now do is set up tube C, which is going to give us an idea of colour change so that we've got something that we can compare to. Because in this investigation, although we are looking for a colour change, we are not using a colorimeter. So we are judging it by eye. So there's a lot of room for human error. That's why it's good to set up a tube that we can compare to. So we're going to set one up. We're going to allow it to go to completion. So we're going to allow the DCPIP to fully reduce and lose all its colour. And that should leave us with a tube where the colour in it we know is the end point. So we can compare all our test runs to that so that we know that they have definitely reduced in terms of the DCPIP. So setting up tube C, which is going to be the one that we are going to be comparing to. So again, we're going to label it so that we know which tube this is. And this time we're going to put 5, 1 and 1. So 5 of the DCPIP, 1 of the water and 1 of the chloroplast solution or extract. So 5, dribble it everywhere. Okay. Only 1, so we can go back to using this smaller syringe. 1. Leave that in there. And then 1 of our lovely green chloroplasts. Okay. Make sure, give it a gentle little swirl so that we get some mixing, and we can hopefully leave that to do the dough. So you can already see at the beginning that it is quite a rich blue, but hopefully that blue will start to disappear as the DCPIP becomes reduced, because this is having light hit it. So hopefully we have got chlorophyll, got excited electrons, we've got photolysis of the water, and we should be getting the reduction of the DCPIP in here. So we can compare to that. Whilst we're doing that, we can leave that and we can consider how it is that we are going to investigate light intensity. So what we are going to do in this practical is we are going to measure 2 different light intensities, and the way that we are going to change or operationalize our variable is that we are going to change it by changing the distance. So I've got a ruler here. What we're going to do is we're going to measure 2 different distances. We can take repeats if we wish, but we are essentially going to place the lamps at the light source 5 centimetres away from one of our test tubes, which will obviously be a higher light intensity because the light is closer. And then for the second one, we are going to have the lamp 10 centimetres, so double the distance away from the light source. So that should make the light intensity lower. And then we are going to use our stopwatch to time how long each one takes to decolorize, and hopefully that will give us an idea of how the light intensity is affecting the rate of photosynthesis, so the time taken for the DCPIP to reduce. Okay, so now we've left tube C for long enough that it has finished. Now hopefully you can see this, but it is very much green now rather than blue. So if you look, there really isn't much hint of blue left. I don't know if this helps at all. Okay, so we know now that that has fully reduced the DCPIP, and that's going to be our comparison now for all the other ones. Okay, if you consider how dark, I know this doesn't have any chloroplast in it, but how dark and rich the blue is, we have definitely lost that blue. Okay, so now what we can do is actually start to investigate the effect of the light intensity. So what we've got here is a desk lamp and a ruler. So what I've done is I've made a mark on the desk using a marker for 0, and then we are going to run our first investigation at 5 centimetres away, so distance. So we're going to set up the rack at that distance away. Okay, so we'll do them one at a time. So this is going to be our first one. So we're going to label the tube X, and I'm going to also write 5 centimetres on it so that I know that that is the correct tube. So what we need to do now is make sure that we are putting the correct proportions in. So we're going to put 5 of the DCPIP, 1 of the water, and then we're going to add that 1 centimetre cubed of the chloroplast solution or suspension, mix it, and then start our timer. So we do need to be quite ready with this. Now I'm just going to take our little control one and put it in another rack just further away, because we don't want to really blast this with light, just in case we have got any cracks in our foil. So I'm going to pop that over there safely. These ones, doesn't really matter at this point. That shouldn't be doing anything because it has no chloroplast in it. This one has already reached the finale, as it were, so it's already completed all of the reduction of the DCPIP. So we're going to place this tube here, and as I say, I'm going to get it in position. So I want it 5 away. So if I line up the tube correctly with the 5 mark, make sure that this light is shining on it fully. I'll move these ones to the back. Okay, and then we will start adding them in. Now, things to be aware of, obviously, it's a practical, we're in a lab, we shouldn't be eating, we shouldn't be drinking, but also DCPIP can be an irritant to skin in the right concentrations, but also key thing is eyes, so we should be wearing eye protection throughout. So we're going to have our 5 of the DCPIP. Obviously, if you get any spills or splashes on your skin, you should rinse them off straight away. 1 of the water. Okay, and then these next parts, we need to do them quite slick or quite quickly. So we're going to have to start. We're going to have to also simultaneously hopefully add the chloroplast and switch on the light so that we can really try and capture the right time and be as good as possible. Now, ideally, we'd be doing repeats of all of this. If you were here and we were doing it as a class, we would be sharing our results. So, chloroplasts. Hopefully I can get this to work properly and also switch on the light. So here we go. Start. We'll mix. I'm not the quickest. Okay, so what we will do now is we will wait to see how long it takes for it to match the colour of this one. It is already getting pretty close. Okay. In fact, I would say we are there. Okay, so you can see if you agree with me. So this is our control that we know has worked, and this is our test here. I think they are pretty much the same. Obviously, you can pull this investigation to pieces in terms of judging things by eye and not using a colorimeter. Okay, and the idea being we would repeat this multiple times. So that's our 5. Okay, so now we're going to try it this time at 10. So using the ruler, we need to take it backwards to about, I've marked this all on the desk. Okay, so I'm going to keep my control there for comparison purposes, and this is going to be my second investigation. So this is going to be 10 centimetres. So this should be a lower light intensity, and hopefully means a lower rate of photosynthesis and a slower reaction. So again, we've got our 5. I could do with a glamorous assistant for this. I don't have enough hands. 1. Okay, I'm going to mix that. Okay, and then here we go again with the timings. So let's hope I can get this right. I'm going to reset my timer. See how good I can get with this. Right. So we are in start on. Little mix. I know that moved it slightly closer. All sorts of errors in this. Okay. So watching carefully for the colours to match. And in fairness, you could also compare it with that one as well. We're getting closer. Still got quite a bit of blue in here. What a difference 5 centimetres makes. Okay. Still got some blue. I'm hoping we can do this in real time rather than me having to pause this and come back. Okay. We're getting much closer now. Now, an interesting side point to all of this. This is not an energy efficient bulb. In fact, I'm not even sure if I can touch this. This lamp is generating an awful lot of heat. So do consider how this may be affecting the reaction as well, okay? Ideally, you don't really want a light source that is also generating heat, because we know that obviously heat will affect the rate of reaction as well. So these are all things that you could list as confounding variables, things that we haven't been able to control adequately. Okay, I think we have reached that point. Stop. Okay, we'll see if you agree with me. So if we look at these two, I think we can agree that they are both a similar level of green, but as I say, we know that this isn't the best way to judge. Okay. So those were our two different distances. So that was a high intensity at 5 centimetres and a low intensity at 10 centimetres. That's how we have changed this. We've tried to keep everything else the same. We've used the same concentration and volume of DCPIP. We've used the same volume of water. We've used the exact same sample of chloroplast suspension and the same volume of it. We've used the exact same lamp, same source, so it's giving out the same amount of heat to both of them. And obviously what we would ideally do is take repeats so that we could calculate a mean and so that we could identify and discount any anomalies that we might get in our results. Okay, and we've kept things cool throughout, and the chloroplasts are also they're suspended in a solution that is cold and isotonic. Interesting. So consider the reasonings behind that. So the last thing to do then is to come back and look at our control. So we're going to turn this light off. Okay, and we've got tube B. Absolutely no reduction of the DCPIP, no colour change whatsoever. So we know that it was definitely the chloroplasts that were causing the colour change. Okay. Tip those slightly, okay, with chloroplast, without. So that has proved that for us. And then this one, hopefully, I've managed to wrap it up effectively. This is to show that it was the light that was causing the change. And yes, so this is still a lovely blue colour. So without light, it also did not change. Okay, so you've got your variation there. We've got with chloroplasts, but without the light. We've got no chloroplasts, and then we've got a lovely green sample, which has got the chloroplasts and was exposed to the light. Okay, so that confirms our results. And hopefully that's all made sense and you've been able to follow through with it.